today's video, we move on to the concept of reconstruction or building these biological networks, and as the first network we will look at GRNs or gene regulatory networks. I will give you an overview of some different algorithms that exist for reconstruction, but we will not go into the details, and I will also give you some details on the concept known as synthetic lethality. So, what is reconstruction? It is the process of integrating different data sources. There are a lot of different types of data sources. There are actual biochemical experiments, there are several published manuscripts, papers on these kinds of topics, there are other existing databases and so on. So, how do you integrate data from all of these different sources to build a single network? This network basically catalogs all kinds of events, biochemical events that underlie any cell, right. So, you have, so a cell is nothing but a sort of a concerted mixture of all these kinds of networks, right. So, there are several chemical events, you may have signaling, you may have gene regulation, all of these involve finally at the basic stage some level of molecular binding, unbinding, recognition and so on. So, this essentially involves integration of information from a parts catalog. So, the parts catalog is nothing but your genome itself, right. Your genome has all the parts, you can identify all the genes, a lot of proteins from the genome, but then how are these wired together, how are these parts tied to one another, that the process of if you will edge inference is reconstruction, and it is typically the first step towards any modeling exercise, particularly the kind of modeling we looked at in the recent lectures. Of you construct a network, then you study several properties of the network, what is the degree distribution, what is the characteristic path length, how does my network behave, so on and so forth, right. Let us now first start off with gene networks. So, the expression of a gene, if you recall your biology, is controlled by other genes, right. There are transcription factors that will bind to DNA, that will help or prevent the binding of other proteins, polymerase, so on and so forth. So, you have a specific flow of information in biology, you have DNA to RNA to protein, and this process is facilitated by several enzymes and several other helping molecules such as your transcription factors. And the regulatory relationships between these kinds of genes form very complex networks, and these are usually elucidated through various kinds of experiments like chromatin immunoprecipitation, ChIP-seq and so on. Most important thing is these transcription factors will bind to particular places on the DNA, right. So, and these are sequence motifs, right. So, they will bind to an AT-ATA, something of that sort, a particular sequence stretch on the DNA, and reconstruction involves identifying all these regulatory relationships. So, who interacts with whom and what is the nature and strength of this interaction. So, it is essentially trying to identify the edges, the direction of the edges and the weights of the edges. So, these are the three things that you want to build in any reconstruction. So, how does regulation of gene expression work? It is primarily mediated by transcription factors, which are essentially other proteins or other genes as we will generalize, which bind to specific nucleotide sequences and affect the transcription of one or more genes, right. You may have one or more genes in an operon which will be affected, right. And there is a lot of information that has been accumulated about these regulatory interactions between the TFs and their target genes in different kinds of organisms. These are available across different databases. There is transcriptionfactor.org, which is a particularly popular database, and we will look at different types of these gene regulatory networks. The most commonly talked about would be the transcriptional regulatory networks, which involve just the transcription regulation, right. So, if you look at the flow of information in biology, you have a gene, let us call this DNA, this is transcribed to mRNA, translated to proteins, and then you have post translational modifications where your proteins could be phosphorylated, acetylated, and so on and so forth. Typically phosphorylation is the most important operation, right. So, then you must have heard of the central dogma and so on about how information flows predominantly in one direction, but occasionally can also flow in the opposite direction, right. You can exercise control at any of these steps, right. When you exercise control here, that is transcriptional regulation or TRN. You can also exercise control in any of the other steps. This is not that common, translational. This is very, very useful. This is used for rapid on off. Phosphorylation is an excellent switch, and this is the most common device, if you will, in signaling, as we will see shortly, right. So, transcriptional regulation essentially looks at this part, and we will look at other concepts such as how there is amplification that happens in the response and so on at a later stage. But in general, there are various levels at which a cellular network can be regulated, and particularly important is the first part, which is transcription. So, how is gene regulation regulated, gene expression regulated, right. Regulation of gene expression is basically mediated by these transcription factors, and we mostly look at transcriptional regulatory networks which include only regulation through transcription, but remember that this is but a small fraction of mechanisms by which variations in gene expression can occur. What are transcription factors? Crudely, they are genes or proteins. They are actually proteins, but you can generalize them to genes, as we will see in a moment, that control which gene is turned on and off, right. They can either influence a particular gene positively or negatively. It can activate the transcription of another gene or inhibit, repress the transcription of another gene, and there are many databases such as DBD, Jasper, Transfag, Regulon and so on, and these are some statistics which are for Homo sapiens or humans you have nearly 3000 transcription factors, whereas in E. coli you have less than 300 transcription factors. But the thing is, if you look at a gene interaction, gene regulatory network, you will find that most of these transcription factors are hubs. They will influence the function of several other proteins, right. There will be a distribution, but in general you will find that they influence several other proteins. So, what are the different gene networks within a cell? So, typically you find all these terms being used in various papers, and you will have to wrap your head around these. So, you may find all of these are essentially different nomenclatures for gene regulatory networks. So, you may call them gene regulatory networks, gene networks or gene expression networks and so on. But more specifically, so if you ever write a paper, you should try to make sure that you carefully delineate what is it that you are talking about. Are you talking about some just genetic interaction, or are you talking about transcriptional regulation, or are you talking about co-expression, right. So, you can imagine that gene co-expression network will connect genes that are expressed together under one condition or under many conditions. Genetic regulatory is a little more generic term, transcriptional regulatory is very specific, right. So, genes that transcriptionally regulate another gene will find their place in this network, and then genetic interaction networks. So, let us look at a simple biochemical network here. So, you have a bunch of genes. So, you have gene 1, gene 2, gene 3, gene 4, this is what you can call this the gene space, and then you have the proteins that these genes encode for, followed by a metabolite reaction, an enzymatic reaction that protein 2 catalyzes. So, you have, you can think of these as either genes or their mRNAs. So, what are the different interactions shown? The regulation of gene 2 by the protein product of gene 1, the regulation of gene 2 by the complex 3,4, which is in turn found by gene 3 and protein 3 and protein 4, which in turn comes from gene 3 and gene 4, and then the regulation of gene 4 by metabolite 2, which is in turn produced by protein 2. Protein 2 is the enzyme that is involved in the production of metabolite 2, which has some sort of an effect on gene 4. It could be an allosteric inhibitor or whatever, and so on, right. So, these are the different kinds of behaviors that you see here, right. So, what is the gene network now? You will have to project all these interactions that happen in the metabolic space, the protein space into the gene space. You finally want to draw something like gene 1, gene 2, gene 3, gene 4, and you want to basically say something like this, something like this, something like this, and so on. What is it really? It looks like this. So, you project all these interactions to the gene space, which constitutes the corresponding gene network, which will look somewhat like this in this case, right. But how does it differ from many of the other networks we saw? There are self interactions, right. That is because most genes will have a negative effect on their own concentration because of the mRNA degradation rate. So, higher the concentration, faster the degradation. So, how many nodes? Each gene is a node. How many edges? You normally would have had n choose 2, instead you have n squared here, because the diagonal can also have, so the diagonal will carry all minus 1s in this case in some sense, right. Or in fact, your numbers in the matrix can be any number between 0 and 1 or minus 1 and 1. Minus 1 would be perfect repression if you just consider a normalized case, whereas plus 1 would be perfect activation, 0 is no interaction at all, right. That is how your matrix might look at for a gene regulatory network. So, so basically the level at which a gene is present is going to higher the level of a gene, it is going to get degraded faster.
Transcribing very fast.
No. So even though it is trying to transcribe very fast, it's going to, the mRNA is going to degrade very fast. So because of that, the rate of the protein is going to come down. So it will be at a particular range, right. So the higher the mRNA, higher the protein production, but faster the degradation. So it will quickly, so it will kind of pull to a sort of intermediate state. It's like a negative feedback. Right. So this is how the final network looks like. So gene 1 inhibits gene 2, that's because you project this, right, and gene 4 activates gene 2. It's actually through a complex mechanism or whatever, but still you just project it to this gene network. You want to finally build these gene networks and study them. So this ending with the minus sign is inhibiting, ending with a normal arrow is like activating. That's in somewhat in tune with the SBJN that we discussed a few lectures ago. No. So every protein, every gene is, we are assuming, is corresponding to one protein. You may have complexes, in that case you will have a little more complexity, right. So here you have complex 3, 4, right, but that is actually the complex 3, 4 is going to have a function. So together 3 and 4 have this function. So you basically have these two edges, right. So 3 and 4 are, you may want to actually split this by putting one more intermediate node and then connecting it, right, because if you remove 3, then the action of 4 is also going to be affected. These are not truly independent edges, but you will always find some sort of such approximations that exist in any of these networks. You can't really embed the entire interaction truth into a sort of network formalism.
Genes will not be the total number of nodes, right?
Fair, but it's the information that you encode, right? What is it that you want to use it for finally? There are no rules, right? There are no rules, so you have to make sensible assumptions. If some of your assumptions are too simplifying, too detrimental to the modeling exercise, fix them and move on. So how do these transcriptional regulatory networks look like? What is the structure of these transcriptional regulatory networks? So obviously we can cast them all onto a directed graph, where every node is either a regulator or is being regulated, a target, right. So it's essentially a multi-layered network, and you will see how this maps well back to what we studied in the previous classes about motifs and communities and so on. At the basic level you have edges, right. You have different transcription factors there and their downstream targets, and then they are organized into motifs, which makes sense, right. You have a few edges that form motifs. The motifs themselves cluster into independent transcriptional units, right, or different kinds of modules, right. Then finally at the top level you have a really complex regulatory network with a particular distribution of these motifs, the interactions and so on, finally orchestrating this complex regulatory behavior that you find in any living organism. So how does this look? This is your edge. So you have a transcription factor that signals that basically affects a single binding site, and then you have motifs, right. So I have a feed forward loop, multiple input, single input and so on, right, and then these motifs are organized into more complex modules and finally into a really complex network, right. This is the diagram that you are more used to seeing, but if you dissect this you will be able to find such interesting modules and finally some motifs. Of course underlying all of these is individual regulatory interactions, and this is where the biology becomes very important. Now some biologist has done a lot of work trying to identify these edges, but this is something a biologist may not be able to see or analyze, right. So this is where the different behaviors emerge. This is the final reductionist view, right. You have single interactions, all these single interactions together make up for a very complex network which cannot be understood just in terms of these simple interactions. You know, go back to the first class where we talked about the 7 blind men and the elephant, right, because if you only look at this part of the network you may not be able to identify many of these interesting crosstalk and behaviors, and this becomes very crucial in any network. The moment you say network, crosstalk and interactions, cross interactions become very crucial, but more so in a biological network where the networks are really large and you have such very interesting emergent properties, right, that you do not find in a smaller network or when you have such isolated networks. So this is one more reason to say why we need to look at a systems picture to study these complex networks. Inferring gene regulatory networks is a very big field of study in itself. One could have a full course on inferring gene regulatory networks. The usual goal in inference is elucidate what we call hidden regulatory events. It is just another way of saying let us say these are all my genes, more importantly I also may have such hidden events, right. Hidden edges, right. So these edges may basically not exist or you may not know them ahead of time. These may be experimentally elucidated interactions, but then you may further infer based on how the network behaves and so on that these edges must also exist. So this is the process of regulatory network inference, and this is a tricky process, right, and there is a lot of study by say chemical engineers, electrical engineers, biologists from different fields of engineering and science, but the trick is you have 2 nodes, that is fine. First thing you have to infer, as I was telling you, is there an edge? Then what does it look like? Is it a plus or is it a minus? And what is the strength? It becomes very difficult to infer some of these things. There are many simplifying models, but you know there are many interesting ways to determine some of these edges. We will look at how, for example, it is easier to construct protein interaction networks, but one common task is to construct a gene regulatory network, right. A typical thing you will have is a matrix, right. So this is going to be G1, G2, Gn, and this is let us say expression 1, expression in condition 2, expression condition M. This is a very classic matrix that you will get, right, a gene expression matrix. From this you need to infer essentially an n cross n adjacency matrix, right, where you know let us call this alpha. So, where alpha I j you have to first find out whether it is less than or greater than 0 or equal to 0, right. First of all, whether does it exist, then is it positive or negative, and what is the number exactly. So, one simplifying way that has been studied in the past is you basically write out G I, the expression level of the ith gene, as some weighted function of right. So, you just say that every gene, the level of every gene is some, in this case a very simplified linear function of other genes.
Alpha ij or w?
Yeah, so you can have any, yeah. So, if this is the matrix that you want to infer, then you can say it is equal to alpha ij into gj. Right. So, the expression level of a gene in a particular condition and so on. So, how do you infer this? Obviously, you might have infinitely many solutions. So, one way is to look for sparse solutions and so on. Can you imagine a problem with sparse solutions? A sparse solution would say that basically the function of one gene is influenced by only few other genes, but you know that there are hubs in these networks and so on. So, that may not be entirely accounted for when you go in for a sparse solution. So, how do you infer this becomes quite a tricky challenge, right, but it is typically a data driven inference. So, you have a lot of data and how do you infer the interactions, right. This basically finally translates into this, right.
Always underdetermined.
It's always underdetermined. You see, if this you usually work with so little data, right? So it's invariably very underdetermined.
So gene 1 will have values for m expressions in the matrix.
Yeah.
So
sometimes and m is typically much smaller. So typically, right. So let us say usually a very reasonable number would be, even for say E. coli, m would be 100, this is already asking for a lot, n will be 4000. Okay. So n is the number of genes and m is the number of conditions. Have gene expression under different conditions.
Answer. So what is Gi? That value is from this matrix or calculated from adjacency matrix.
So Gi could be the level of expression of or this could be even in some condition or the average expression of gene I across the m conditions,
okay,
something like that. But then you can say that that is related to in some linear fashion to other genes, to the expression values of other genes. Okay. So you could say something like G1 is 0.3 G2 plus 0.4 G3. Something like this is a very simplified model. You can have many more complex models, but this is a very fundamental simplistic model. There are many other algorithms, I will not get into the details, even it's not we have not even looked at this in great detail, but this is essentially one way to go about assuming how the genes interact, right. We assume that the level of every gene is some linear combination of the levels of some other genes. No, so we basically assuming that the entire network is hidden. I do not know any alpha here. So I solve this equation, right. This is going to be a very difficult equation to solve. So you need to have another axis, right. you could have another temporal axis, right, and then you can use, and then you could even say g1 of t is some g1 of t minus tau and so on. So, you can have any number of complexities here, and this is a very intense field of research. I am not going to get into the details, but basically know that there are many algorithms to reconstruct networks, and these could be useful for your final project and so on if you want to, because abundant gene expression data, right. You have gene expression and RNA-seq data for several organisms today from different types of databases. Okay, there is a gene expression omnibus, which is now essentially the only big database. There used to be Stanford microarray database, but that is sort of now shut down, it is deprecated. So, the gene expression omnibus is one source for huge number of such gene expression data sets. So, you can infer, you can start with clustering the data, right. So, clustering might give you some insights into what is what kind of genes work together and so on, but that would be the simplest form of gene expression data analysis. But then you may want to go ahead and try to use network data or text mining or comparing networks across species, right. You may have some known networks in some species. Can you compare your species with that and identify if there are going to be possible edges that will be conserved from that species to yours and so on. There are many other approaches that are based on mutual information, Bayesian networks, differential equations, and then of course using several dimensionality reduction methods and so on to find what is the effect of one gene by the others, and what are the top 5 genes affecting my gene, something like that. You can start building those kinds of analyses. So, there are some algorithms called reveal, MNI, NCA and so on, which are popularly used. The other interesting set of interactions is something known as synthetic lethality. So, let us consider a cell which has 2 genes ABC and say PQR. When you delete ABC, nothing happens to the cell. When you delete PQR, the cell still grows. When you delete both ABC and PQR, so this means that there is some interaction, genetic interaction between these 2 genes, right. So, maybe they were both producing the same metabolite. If one was there, if ABC was there, the cell could still grow. If PQR, instead, if when you hit ABC, PQR could compensate for it, or vice versa, if you hit PQR, ABC could have compensated for it, but when you hit both, the cell can no longer survive. You can also have higher levels of such interactions, right. So, you remove this, nothing happens, you remove this, nothing happens, but if you remove this also, the cell dies, right. You remove any pair, you remove AB, BC or AC, the cell survives, but when you remove A, B, C, all 3 genes, the cell dies, right. These are higher levels of synthetic lethality. This is a very useful tool to study interactions, genetic interactions in different organisms. You have typically you have some synthetic lethality screens, basically big genetic arrays to experimentally study. So, they are called synthetic genetic arrays and so on, to experimentally study lethality. You can also predict these based on the gene network or the metabolic network and so on. So, in many of these cases we may not actually worry about finding the alphas as such, we will stop at finding if alpha is not non-zero, right. First step is to find if alpha is non-zero, and maybe if possible find the sign of alpha. Finding the value of alpha is a lot more tricky.
So, lethality only exists for pairs?
No, there are single genes that could be lethal. You could have another gene like DEF, which when you delete the organism will die already, right. So, these are essential genes. These are synthetic lethal pairs, and one would call these synthetic lethal triplets. So, there is a lot of studies, we will study some of these later in the course as to how these are studied using metabolic networks. It's quite easy to study these using metabolic networks. Wherever you can predict the phenotype, you can start and the phenotype on a given perturbation. So you remove A, see what happens. You remove B, see what happens. You remove A, B, and see what happens. Okay. Using this kind of a methodology, you can actually identify synthetic lethals in any organism. So all these can help you to form hypotheses, which you can later try to validate through other kinds of experiments. So this is what I just explained. So you get non-lethal phenotypes when you delete a single gene, but when you delete both genes, you get a lethal phenotype. This is called a synthetic lethal because you synthetically superimpose the two deletions. Right? Only then you get a lethal phenotype. So I hope you had a good overview of how one builds and reconstructs networks, particularly gene regulatory networks, and we also looked at the concept of synthetic lethality. This is a topic that we will revisit in little more detail when we look at metabolic networks, because there are very interesting algorithms to identify synthetic lethals in metabolic networks. In the next video, we will look at ways to reconstruct protein-protein networks, the kind of networks you find in string and database and so on, and methods such as Rosetta Stone, phylogenetic profiling, and correlated mutations which enable you to predict these functional associations.